Okay, well, good afternoon, everyone. My name is Ida Fernandez, and I'm the Executive Director for the Imagine Spore Network. The Imagine Network is investigating the interactions between inflammation, microbiome, diet, and mental health in patients with IBD and IBS. And so our mission is really to transform the management of these diseases and the associated mental health issues that is associated with these disorders. So thank you for joining us and welcome to today's session. This is now the 5th installment of our network's monthly training webinar series, and it's really aimed at building capacity and improving the design of future research studies. I'm excited about today's talk on diet and microbiome research, and hopefully we're going to have 2 presenters join us today, but we will see, as one of our presenters seems to be held up a bit. But we'll see if she joins us towards the second half of our webinar. So first we have our Diet and Environment Working Group co-lead, Dr. Leo Dieleman, and hopefully joining us later will be registered dietitian Cheryl Nickurak. First, just a few housekeeping items before we get started. So I'm just going to ask everybody to please keep yourself on mute throughout the presentation so we can minimize any background noise. We'll allot maybe about 10 minutes towards the end of the webinar for some Q&A. So we encourage you just to type in your questions using the chat box at the bottom of your screen. And you can do that at any point during the presentation. And then at the end, I'll read them out to our presenters so they can answer them. Following the meeting, just a reminder, you will receive a link to complete an evaluation. So please give us your feedback so that we can continue to improve on these sessions. And it also allows us to track participation for those that are seeking CanMed credits. Finally, we do record these sessions and we archive them on our website at imaginespore.com for future viewing. So you can feel free to visit the website and share it with your colleagues if they weren't able to join us today. And you can also check out our previous training sessions as well. So before we get started with the presentation, I'd like to introduce you to our presenters. So first we have Dr. Leo Dieleman. He's a professor at the University of Alberta, co-director of the IBD unit at the Division of Gastroenterology there. As I mentioned before, he is one of our co-lead for the Diet and Environment Working Group of the Imagine Network. His interest is really in new therapies for IBD that have also led to the involvement with his, has also led to his involvement with the Edmonton-based company Bioimmuno Designs as its co-scientific director. He's the associate editor with the Journal of the Canadian Association of Gastroenterology and Frontiers in Medicine, and a reviewer for numerous other scientific journals. He's published more than 100 original scientific articles and book chapters in peer-reviewed journals, and is a frequent presenter at international, national, and regional scientific conferences, mostly focused on his work on IBD. So he's got a strong interest in understanding and management and prevention of IBD through diet or diet supplements that change gut bacteria. Cheryl Nickurak, who again hopefully will be joining us later today if she's not already on the webinar, is a registered dietitian and registered nutritionist. She's with the College of Dietitians of Alberta and the Dietitians of Canada. She's a Diabetes Canada Professional Sectional YEG Executive Committee member, and is involved with the SPORE Network as well as a KT workshop attendee. Her focus is on prevention and treatment of inflammation in IBD, nephrology, cardiovascular, diabetes, celiac disease, and allergies. And she's a registered dietitian, as I mentioned, leading a study in diet and ulcerative colitis at the University of Alberta. So I'm going to pass it over to Leo right now to carry on with the webinar.
Okay, good morning, everybody. Thank you, first of all, Ida and the Imagine group to invite me to give this particular seminar. And with the idea is that we are trying to, as a knowledge transfer to all of us participating in this, and see how, why we're doing these kind of studies, or how this actually fits into our Imagine goals of research. So these are my disclosures and Cheryl's disclosures as well. What we're trying to do today is the learning objectives, is we're trying to find the relation between diet and microbiome, particularly focused on IBD, Then I'll lead you into the latest dietary research in IBD. And again, there's so many things happening at this point that I don't even claim to be that is most up to date, because it's happening all the time. And then Cheryl particularly will talk about the benefits and challenges in conducting dietary research, as you can imagine.
Leo, before you start, you seem to be coming in and out with audio. Do you just maybe, I don't know if you want to get a little bit closer to your microphone. It's still okay.
Okay.
Great, that's
okay.
better.
Maybe that's the computer then. Okay, yeah, because I'm talking to the computer microphone.
Okay.
So I don't have a mic over here. That would be better. Okay, I'll be cognizant of that. So I hope everybody you can hear me now, okay?
Yeah.
Okay, yeah. So as I said to you, for the people joining, I mean, most people know what Crohn's disease and ulcerative colitis is, but just to give the highlights for people who are coming from the outside, that ulcerative colitis is mostly confined to the colon, as the name already says. It's a more superficial inflammation. You can see how it distributes, and some people have proctitis. Some people have only left-sided disease, and about a third of them have pancolitis. This is Crohn's disease can actually occur anywhere in the GI tract. It's typically characterized by skip lesions. It goes through the wall. It's transmural. This leads to abscesses, fistulas, strictures, which we typically don't see in ulcerative colitis. And most importantly, if this disease is outside the colon, and particularly in the small bowel and elsewhere in the GI tract, you can think about Crohn's disease. But they all have in common, it is our chronic inflammatory diseases that's where the etiology is still relatively unknown, and there is no actual cure. So as I already hinted to, how the pathology is also very different, and that you can actually see, as I just alluded to, that you see that ulcerative colitis is more like a diffuse inflammation, but Crohn's disease, you can see here, goes straight to the wall. In some of those pictures, you can see that these fissures and the fistulas that are appearing and leads to all kinds of complications. Frequent surgeries, particularly in the past. With our current treatment, we have gotten a lot better. Colectomy is treatable with medications, but again, the colectomy, ulcerative colitis is typically treated with medication, but colectomy happens and still happens in about 20% of cases. So that basically comes to our goals for this particular talk. So once you actually get to a patient who has already the disease, and you can imagine the picture on the right is ulcerative colitis, is very diffuse. The picture on the left is Crohn's disease, but you see these skip lesions, the aphthous, some of these aphthous ulcers or small ulcers, but particularly when it's established disease, it's really hard to find questions about what triggers the disease. You have to ideally try to look in early stages and before disease occurs, looking at early stages of disease, or even looking at first-degree relatives and people who are healthy from Crohn's patients, as we do in the GEM study. So this is the map of how IBD was in the 1960s. And you see there was not much known. There's lowest ranks. There was something about Scandinavia. There were some, mind you, Dr. Crohn, who gave the disease, Crohn's disease, we talk about something in the 1930s, and more people began to realize that this was increasing. Now, if you look at the map, and now you're looking at the map of how it's been increasing, that you can see, and this is for Crohn's disease, how you can see that in the United States, Western Europe, and Canada in particularly have one of the highest incidence and prevalence of Crohn's disease. So now the question then comes, like, why is this happening? And everybody knows it can be, particularly in Crohn's disease, a genetic susceptibility. That genetic susceptibility is also present in ulcerative colitis. But more importantly, we began to realize that the environment is very important. And when we talk about the environment, we look at the role of microbes, we look at the role of diet that can affect the microbes. We know that smoking is worsening Crohn's disease and actually has a relatively protective effect on osteocolitis. And so if you have the combination of genetics, genetic susceptibility, and the environmental factors, that's when you get the disease. This is the old Venn diagram, as you probably remember from mathematics class. That here is you're sitting in this, and when these things come together, where you got genetic factors coming together with when these genetic factors coming together together with the environment, that's where you get the disease. So you can basically living in Africa in a healthy microbiome environment, you could be genetically susceptible. Nothing is going to happen to you until you move to an area where the environmental factors are present, and then you basically move from here to there. And the same thing is true with genetics. If you have just the environmental factor, but you don't have the genetics, then by definition you should not be able to get the disease. These are the kind of things that we hope to solve with the Imagine network. Only large studies with big data were able to we talk about the cohorts that you probably heard about, that we are looking at 8,000 individuals, 2,000 Crohn's, 2,000 osteocolitis, 2,000 IBS, versus 2,000 controls, to get a handle on what these particularly factors are, particularly in the microbes, particularly in the diet, even and mental health plays an important role here as well. So my focus will be on the intestinal microbiome. The intestinal microbiome made the cover of Nature because people realized this was actually one of the most neglected areas of research. Everybody was looking at the host being the human. Everybody knew everything about the human cells, but you have to realize that there are quite a bit more cells in the outside, tenfold the number of human cells that are in your gut. And so that it's very likely that this gut microbiome will crosstalk to the host and as a result lead to health or disease. So you just studying in the gut microbiome are absolutely necessary to understand why certain diseases happen in human beings. So I had the privilege to work with Belfer Sider at the University of North Carolina in Chapel Hill. And Belfer was my mentor in the 1990s, and had a concept which was very novel at the time, that they have a genetically susceptible animal in this way. It's an animal model for colitis, A2A B27 transgenic rat. And once you have these rats have the transgene, and it's the same transgene, same gene as for ankylosing spondylitis, which is a rheumatological disease. So he inherited this model from a rheumatologist who noticed that these rats actually developed colitis besides arthritis. And he said, no, I don't need anything with colitis. And everybody was afraid that this colitis was due to some kind of a pathogen. When we made these animals, he created a germ-free model together with scientists in Michigan, and then noticed that these animals had no disease, no colitis, and no arthritis, and they were germ-free. So there were germ-free isolators at the veterinary college in Raleigh, where I had the privilege to work. And when we introduced bacteria, Sarcoidosis bacteria, into these germ-free rats, they were getting very aggressive colitis, as you could see here. When you introduced one particular organism, this is Bacteroides vulgaris, there was moderate colitis. But if you introduce E. coli as another different species, there was no colitis at all. And if you entered lactobacilli, such as a probiotic, you can actually protect the development of colitis in these animal models. So basically, this was forgot the concepts that not all bacteria are causing disease. Some of them are causing disease, some are protective, and some of them remain neutral. And that was an actually very important concept to understand colitis and IBD as well as Crohn's disease, osteocolitis and Crohn's disease. So the concept that we developed in the 1990s that, the body has that the development to the ability to develop disease is depending on this balance of protective versus pro-inflammatory organisms. And this is very simplified. I mean, this is just introducing concepts that the bacteroides in this animal model were pro-inflammatory, and so were other species like the Enterococcus faecalis, some pathogenic E. coli, as we know, and some Enterobacteriaceae, which is actually more and more becoming the so-called pro-inflammatory pro-inflammatory bacteria. On the other hand, and this side of the balance is the protective. And if you have relatively paucity or like not enough of probiotics, lactobacilli, bifidobacteria, some of the non-pathogenic E. coli became probiotic as well. That's the E. coli nissel. But you've probably heard about the Faecolobacteria prausnitzii that are protective. If you have enough of those, then you protect the development of postoperative Crohn's disease. And then what a lot of these bacteria had in common, they were protective. They were actually producing an anti-inflammatory molecules with a short-chain fatty acids. And I'll come to that concept. But basically, it's the balance, the disbalance that leads to disease. So we call this in the intestinal dysbiosis. But this intestinal dysbiosis is not unique to IBD. It's actually also found in lots of other diseases, as you can see over here, and that's resulting in inflammation. And you can see here that is now being thought in also in colon cancer, but also in diseases that you might not particularly as associated with the gut, such as diabetes, such as multiple sclerosis, heart disease, and so on, and even allergies that we currently see now, and the increase of autoimmune diseases seem to be correlated with the dysbiosis that actually could be happen early in life, but possibly by the introduction of antibiotics early life. So anyway, there's the concept of dysbiosis leading to diseases and leading to inflammation. Now, how does this translate into IBD? And this is an overview by Marcus Neurath from Germany, who put this into in 2014. Sounds already old versus some of the medic, but I thought he did a really good slide here. And so what the current idea is the development of IBD is that you are genetically susceptible to your genetic factors. We have several of those genetic factors, up to 170 of them. But they all have in common is that all the genetic factors seem to have to do with the way that bacteria handle, that the immune system handles bacteria. Then you have your environmental factors, your microorganisms, your diet, and here it is. But also something like the effect of smoking, as already alluded to, and even the use of NSAIDs, which are the non-steroid anti-inflammatory drugs, such as ibuprofen and Advil and naproxen that people are discouraged to take. But it also, and this is a concept that is becoming more and more important, that it seems to be also with impaired barrier function. So if your barrier for all those bacteria is leaky, then you can see how things are getting through that are mostly bacteria, but also dietary components. And the immune system that will have a response to it. The immune system will then get overwhelmed with a pro-inflammatory response. And then on the other hand, if you also have a genetic susceptibility for the failure of the regulatory mechanism, you have disbalance in the immune system for pro-inflammation, which is the one that are trying to block it. If that balance is also off, then you can see how chronic inflammation occurs because bacteria getting through, inflammation keeps on going, and you need the anti-inflammatory drugs. But that part is all, the current drugs are all in this particular area. What we don't have therapies for as much is this part, the environmental factors. How do we do anything about that? So I set up a model, this HLA-B27 rat, they're transgenic and they develop inflammation. So you can see on this slide here on the left-hand side, This is a very inflamed gut. This is a cross-section from the cecum, and you can, it is full of inflammatory cells. However, when we gave a fermentable fiber that's called oligofructose inulin into the drinking water of these animals, and we can actually see this really reduce the development of colitis. You can appreciate that this layer is much thinner than the layer over here. You don't see as many inflammatory cells. And this was like a remarkable observation. I know about probiotics. I knew about Karen Mattson studies on probiotics and Dr. Fedorak. I just wanted to do something different. How can we actually stimulate the good bacteria in the gut with a fermentable fiber? And that's actually what prebiotics are. So when we did see that, we could correlate an increase of bifidobacteria and lactobacilli. So what I was, remember the balance that I showed you before? You see now that the increase of those bacteria actually occurred in this model. Everybody said, okay, now there we go, we got it. It's just because now you stimulate the so-called good guys versus the bad guys. It's very simple. Well, we translated this into, so, well, it's nice to do this in animal model. Now we do translational research. So we use the same prebiotics. The oligofructose was derived from a chicory root from a company in Belgium. Then we said, okay, can we make this into a clinical trial? So we did a pilot study first before you do a clinical trial. And so the problem was in lots of the studies with probiotics that probiotics were given, usually one particular strain, one particular dose. I wanted this to stimulate, I wanted to find a dose, and if I find a dose and a difference in a dose, then I had a pretty good idea in a pilot study what I would do in a more randomized placebo-controlled study that we currently just finishing. But these are the first results, and you can see this is the remission. Clinical remission means you have in ulcerative colitis, these patients had mildly active disease. We gave them the prebiotics, and they were all on their baseline medications, which this is oral 5-ASA. And if you give that on top of these medications, you get 50% of these people in remission, which was a remarkable finding. You can also see here, sorry, my pointer doesn't really work that well, that it's a dose finding. You see less of an effect in the lower dose when you give them 7.5 gram a day. And this was like a treatment of about 9 weeks to do this. And you could actually see in this column that some people were here, they were worsening in the lower dose. Now, it's interesting to find something, but it's more interesting to find why. So if you look at the investigation of the microbiome, you can find out who is there, and this is what you should do with the 16X RNA, the viral sequencing. You can also look at, more importantly, is what is their function, and you look metagenomics. That's much more deep dive into genetics, and then finding out what they actually can make. And then you can look at substances that you can find in serum and urine and stool, what the bacteria are doing. This is coming from bacteria, and it's coming also from the host. So there's several ways, tools that we currently have that we didn't have before in order to answer the questions, okay, this works, and we know it has an effect on the microbiome, but how does it work? So we used the pyrosequencing in this pilot study, and everything, and you can see that the more, so first of all, yes, your ulcerative colitis disease activity score, and that correlates nicely with the fecal calprotectin. So this is a standard test that now we're using in the clinical practice. That the more, and this is fecal calprotectin is a protein that gets made by macrophages and neutrophils. So the higher the number, the more inflammation you have in the colon. And so we correlated, you could see that the higher the fecal calprotectin, then those guys light up in the red, and those are the ones that are pro-inflammatory. Versus the so-called the green guys they are correlating with protection. So low fecal calprotectin, low inflammation. So you actually see that there is what we just showed you. There is here showed the disbalance. You can see how certain bacteria are correlating with beneficial effects of giving these prebiotics. And those what they all have in common, these bacteria, most of those Rhizoborea, the Faeculobacterium, they're all producers of short-chain fatty acids. So prebiotics, fermentable carbohydrates, your body cannot, it doesn't have the enzymes to break them down, but the colonic bacteria do have several metabolic enzymes to use it into fermentation process. And this fermentation process leads to the production of, and that's just over here, of the those short-chain fatty acids. Those are the main ones. We have, but the most important one is butyric acid. Butyric acid is one of the fuels for colonocytes. If you don't have enough butyric acid, your colonocyte will become leaky, or you get colitis, as you can actually see in people that have a ileostomy and they have a diverted gut. And that diverted gut, who doesn't see bacteria, gets actually inflammation. That's due to the fact that they don't see bacteria producing butyric acid. And butyrate is also important as a regulatory molecule, is an anti-inflammatory, as I said. So here you have production of natural occurring anti-inflammatories in your body. So if you don't stimulate those bacteria to make that, you can actually get disease. So, and as a result, but the side effect is also of fermentation, you produce gas. So if you give too much of it, these compounds, and these prebiotics can also be found in foods, by the way. You can find in garlic, onions, artichokes, and as you eat a lot of them, you can get very gassy and bloaty. And that's actually the limitation, is you can eat too much of them. So that's a side effect. But anyway, using that, we actually said, okay, is there a difference in the butyrate production between the ones that are responding and non-responders? And lo and behold, we did find that. So it's a very nice correlation that butyrate is a predictor for response. So here we have a mechanism. Yes, it's the difference in the beneficial bacteria are being stimulated by prebiotics, and they also they produce butyrate. So this is a function of the bacteria. Now, coming to the diet, this was the first kind of, call it dietary supplement study, but more important is what is the overall diet doing? So this is an important slide. It's one of those landmark studies. This published in 2010 in the Proceedings of National Academy of Science by De Filippo. And what he did in this is he went to an African country, Burkina Faso, and compared the gut microflora with the ones for the European settings. And what you can see, although you can see that there is both Bacteroides and Firmicutes, but the percentages of Firmicutes and Bacteroides are very different in these populations. You can see the green are the Firmicutes, and the reds are the green are the Bacteroides, the reds are the Firmicutes. And if you look this over time, and particularly who is there, you can see that just by looking at the slide and the colors, you can see how different the populations in the gut bacteria are between those two populations. Well, we know that we eat very different things in our society, Western society, versus Burkina Faso. And so most of the studies that we've done, you see this is not new. This is something that came out already in 2004, where people were not paying too much attention at first. Now it's a hotbed of publications. But the concept here is that our Western society has an increase of animal protein and fat, and particularly refined carbohydrates. Think about all the pops that we have, versus a relatively decrease of whole grains, fruits, and vegetables. And that the dietary change, according to some people, could be responsible for the increase of those pro-inflammatory chronic diseases such as IBD, such as allergies, and you name it. Just what I showed you on that other slide can be actually inducing dysbiosis as one of the factors. So we did some studies. This is a very small cohort study. We had this medical student from the Netherlands studying this particular thing together with Amar, one of my PhD students, and looked at people that actually had ulcerative colitis, that there were relapses in the last a few months versus the ones that stayed in remission. Interestingly, in this very small study, we saw that the increase of monosaturated fats was actually correlated with certain fats increase of relapses. So in other words, there was some indication, and some more studies are coming out, that relapse frequency and also colitis seem to be correlated with the diet. So that brought us into the concept of developing the diet study. We said, okay, I think we have enough evidence that we need to study the diet. And so we developed this together with the diabetes group we had with Rhonda Bell and Kendall van Diepen, one of the dietitians. And then with help of Cheryl Nickurak, this would be my next speaker, this study where we said, okay, patients in remission of ulcerative colitis will have flared less than 18 months before are at risk for the next flare. We began to understand that in order to make a real impact, you probably had to go to the patients in remission. So you see the anti-inflammatory diet, and then the control had to be the Canada Food Guide. So ethically, it's not permissible, permitted to let people just go on their own and say, okay, well, you will not be blinded if you do that, if you don't give them an option of being in a dietary intervention. But of course, the anti-inflammatory diet was what we really wanted to aim at. And we did face-to-face dietitian counseling, and that's why Cheryl's role became very important, to make sure that people were compliant. And this study was going on for 6 months. This is the longest diet study that is done in real life that has been performed so far in IBD. And in the meantime, we're looking at, okay, can we prevent flares? And so we have clinical parameters like having blood in the stool, frequency of bowel movements is the partial Mayo score, but also looking at inflammatory markers in the stool, as you showed you, the fecal calprotectin, and other things. So here we go, the anti-inflammatory diet. This is what we're trying to aim at: increase of the prebiotics, less sugars, emphasis on omega-3 fatty acids, such as found in fish oil, olive oil, and then particularly a decrease of the intake of red meat and processed meats, and particularly sugars. So when we did this, and after 6 months, we're extremely happy to see that actually we were able to, first of all, this is a slide, we used an anti-inflammatory diet. So let's see if a validated diet score, such as the Dietary Inflammatory Index, this is a score developed in South Carolina. And then you can see how, if you're using this and using at the stuff that people eat, it can be then see, are they compliant? And you can actually see that although they both were showing an improvement, that both groups were in the dietary index of about 0, which is not too bad. But it's the anti-inflammatory diet who went further down. Just the more negative you were, the more anti-inflammatory you were, versus the control group basically stayed where they were. That was not significant. And this was a big kudos to Cheryl, who had been sitting on those people for 6 months and make sure they were compliant. So that concept worked. So it can be done.
Leo, just a time check. We're at about 12:36 right now, okay?
Oh, sure, yes, okay. So anti-inflammatory diet did prevent colitis, but it was the fecal calprotectin, you can see it prevented subclinical inflammation. So, and you can also see how this nicely correlated with the metabolomics. So metabolomics is the study where you look at bacterial products, at dietary factors, and you actually could see that the people that were in the anti-inflammatory group were segregating themselves out from the baseline. So, this was not the only diet study done. Eat and Wine also part of our group, and here at University of Alberta, together with Ari Levine in Israel, did a diet study in children. Basically, kids were giving, but the difference there is that they took out pro-inflammatory factors that are found in, like I said, animals. It's more like how much can we take out that we know can be causing inflammation, such as emulsifiers. Carrageenan is one of them, red meats. And so these kids are getting when they're having Crohn's disease, they usually get in the enteral nutrition, which is like can't think about the name now. It's as modulin, and so actually the concept is here. So you have modulin given to one group, but the problem with that is the modulin is group 2. This is a control group. It's very nice. It's not very palatable, so you're not able to take that on for months and months. So it's if you let them eat a diet, a normal diet, but you take out the inflammatory factors, and then let them gradually step down, then you actually have more palatable. So the diet is then better tolerated than just a enteral nutrition. But now you let them. You could this a free diet means you can let them eat after that. But what you can see, and the basic concept is that also in this study, you see a significant reduction of that inflammatory protein, the fecal calprotectin is in here, versus the group that were not, the diet was not restricted and were eating. You see actually now this number is going up. So here you get a so-called sustained remission in the group where you actually let them eat a diet that is controlled and devoid of pro-inflammatory factors. So what does it do to the microbes? That's a really important question. The hallmark is, here are the bad guys. This is the enterobacteria, proteobacteria. And you can see actually that the proteobacteria from one diet to another one, that there is in this, you see that the proteobacteria is really hardly any change. You see that the bad guys are still present, but here the proteobacteria seem to be less in the group that had the diet in the kids. So we see that there is definitely a sustainable decrease of that population. Anyway, so what I want to tack here is that diets is an important thing to look at. You'll see more and more dietary studies, both epidemiologically as well as intervention studies, because the diet can affect something that we can change. We can change the environment, we can turn this disease around. And not only turn this disease around, but many other diseases that are related to the so-called dysbiosis in the gut. So I think that diets can affect the gut microbiome, can affect the role and fine-tune microflora changing therapy for the future. Something that Imagine Network plans to do in large studies, but we do it in smaller studies with pilot studies, and we just finished a randomized control trial diet intervention here at the University of Alberta. So anyway, now's the time for me. Hopefully, Cheryl, I hope you're on the line. And that you
yes,
can,
I am. Do you hear me?
Ah, there you go.
Oh.
Okay, so let me know when I need to move the slides. So this is your part, and then hopefully we have time for questions. Okay?
Okay, I'll do this finger, okay? All right, is there a big echo or do you hear me okay? I don't have a choice. We're in a big room. I'll try to put some coat around me. Okay, so thank you everybody. Thank you, Leo, for inviting me, and Ida and all the group for setting this up. Here, this is the coat. Is that better? All right, sorry about this. Okay, so I was involved with the ulcerative colitis diet study, as we called it. And thank you, Leo, for the big intro there, and Ida. So my role here is to point to issues that we had, or concerns, or research stumbles, I guess I would say, challenges. All right, so we had at month 0, we had an issue where, not an issue, but we had the folks do a food frequency, and 51 out of 53 or 54, we're always debating whether we had 53 or 54, folks did that successfully. And they were mostly handheld to get that process done, if you want the truth, okay? So that was great. And the challenges of the food frequency is that the Canadian version was not up to par. And so Amar manually transferred everything to the US version eventually. And that worked better. And so every participant did the ASA 24 food recall, 1 day recall at month, as noted, all of the months, month 0 all the way through to month 6. And month 12 was looked at but not analyzed by myself, okay? So we looked at, or I looked at rather, all sorts of things within that, but mostly it was the program and more looking at those food frequencies that came in. I looked at their manual version and went from there. So there's several downfalls to that version of ASA 24, which was that yogurt was sort of a 1990 version of yogurt, and that's it. There was no seafood diversity, spices were not accounted for. So there were a number of pre-probiotic containing foods that were not at all on the program. So it was very difficult to deal with. Plus, it was an annoying program to work with. The new version, which we were waiting for at the time, is out, and apparently it's a much easier version. The details of the study were entered into RedCat by Amar. Okay, and my suggestion for the future is other steps is to cut the in-between and have dietitians center those steps. And there's that finger. Next slide, please. Okay. Leo, are you controlling this slide? Oh, sorry. I can't hear. Oh, there we go. Thank you very much. Okay. All right. So month 0, 1, 3, and 6 were face-to-face. And as for most studies and most clinical procedures, we have a pre-assessment where I would also look at the blood work that was done, and then pretty standard lab data. And I want to pick out any information there that needs to be dealt with immediately. Meaning we had about 2 to 3 participants who had diabetes type 2. So I just needed to check those levels to make sure they were safe. I did anthros at each of those face-to-face, height once, but the thing I checked, another stumbling, is to make sure that your height device is actually accurate with another measure to counter check that, because some devices, particularly automated ones, may not be calibrated properly, may not be able to be calibrated properly well. Weight, all those you can see. We also did hip, waist, mid-arm, and calf circumference, so that it could be used later if needed. And so far, those numbers are quite stable at that point. Eating patterns, as any dietitian would, as well as particularly good information like, do you have a garden? Do you use garden food? Pollen food allergies are a standard ask for me, and I'm not diagnosing, I'm just noticing patterns. Of course, seasonal patterns, and also sleep. Do they sleep? Does this person sleep? Triggers, vitamin, minerals, of course, the gamut. Instruction at month 0 were Canada's Food Guide. They were given the older, if you can remember that version of that, Canada's Food Guide, which has the portions on there, or the Alberta Anti-Inflammatory Diet binder, but only 2 weeks of it, out of a total of 4 weeks initially are given. So went through serving sizes, substitutions, a bunch of information regarding how to manage its diet, which can also is the same calorie level, but again adjustable for up and down calories as needed based on the individual's needs. Not in detail, but calories were not looked at or not assessed in detail, just a matter of weight changes and hunger. All right, next slide, please. Thank you. So at each, the telephone sessions, I was in charge of reviewing partial meal with participants, stressors, which you're all used to, and then nutrition changes, weight changes, symptoms, and intake in and out. Eating out, rather, any alcohol changes, challenges, and review 24-hour recall with them before or at the time of submission, and question time. The food guide folks check adherence based on the food guide portions. So to their needs again, just seeing if they're having 2 to 3 milk products, having their 2 to 3 portions of decapart-sided meats, and as well fruits and vegetable portions. So the food guide, which is the next slide, I think. And sorry, inflammatory, just back one, if you don't mind, Leo. And then anti-inflammatory group is going through some details there. I'm not sure why I said 52, but many patients, 53. There were a total of, as mentioned, two vegetarian folks and 1 lacto. When folk carrying vegan tendencies, the big take home with this is that the folks that are using almond beverage, soy beverage, coconut beverage, a reminder that those almond and coconut are 1 gram of protein per cup versus the 9 that cow's milk provides. Lactose-free milk, quite a few had lactose intolerance tendencies, clinically or otherwise, but made use of that product. And I would also go through details of where to buy and what a good, where the sales are, because I know that for myself. So, and what times of weeks and days the sales are on. So they appreciated that. There is one folk who was on the GAPS diet, and that's a whole other session, as well as the nickel-free, which is the same person on nickel-free and GAPS. This is encouraged by the person's naturopath. Okay. So how many games? So we'll just skip right to the chase here. So 75% of the participants did their own 24-hour recalls, entered into ASA 24, and with struggle some of those. The program is not too friendly, and then the other portions were entered by myself and a nutrition student. 14 were not recorded, not done, and this is probably can reflect on stress of those 2. What my big focus was as well is that I made it very clear with the participants is that this study, we do not want it to be a caveat of stress. We do not want that. So however I can help is going to be a better result than you experiencing undue stress due to a study that is not supposed to encourage stress on your condition. So very important. So food frequency, 94% were done, not bad. Some mostly assisted. Okay, languages, English, me, and a few Tagalog, Farsi, and Punjabi. Tracked food intake with the app. I found that those that were more apt to tracking their food intake, mostly the athletes, then they had very little to no problem with entering 24-hour food recalls whatsoever. In fact, they were champions and easily done. And those particular people, as it turned out, had just or recently finished their master's degrees in sciences. Okay, as it turns out. Hey, people, oh, this one back, if you don't mind, Leo. Oh, stuck for time, okay. Thank you. Okay, so of the Alberta inflammatory group, then there were some just problems with, could you just mind going back one slide? Sorry, Leo. So one person out of the 30-something, okay, on the Alberta inflammatory diet actually did the rule-by-rule meal plans for 2 weeks and then wanted to bail on the whole entire study. So that was a good hour on the phone. It turns out that when I was speaking with the patient, of course, it's the spouse that does all the cooking and wasn't involved with the week or the month 0. So that was rectified quite quickly. Okay, so what I like to do with all folks is entertain or carry on my grit ideas is to help clients through is express gratitude, responsiveness, intuitiveness, and teachability, that I am teachable as well. And of course, those sort of principles come through as well with any sort of patient-oriented research for sure. Okay, and next things. Okay, so some of the items that are important for important to note is that these folks went on holidays. 3 of them got married during the study. They had dance competitions, sport competitions. They had real life going on. And you know for yourself with ulcerative and IBD is that it is internalized big time. The stresses are huge. And as well, they went traveling. So I had email appointments, Italy, Denmark. You can see the list of folks, list of countries that Andrew Tim knows from. I ran a stool sample. He forgot. I had to go get that because I had the car. So you got to go get that stool sample and get that to the fridge. So that was me. Okay. So important is that dietitians are a good investment. We know about food nutrition, so it's important to have that going out too. We had one or two members who they all got paid for their parking, but several parking free areas. I know that. So, and they happen to need food. They were low on work at that point, so it went to good cause. So always consider giving coupons if you're giving any for any reason. Would be for food, grocery stores, not for restaurants. We got the rap a few times from a few participants for giving Tim's coupons. So okay, and we're flexible. We had appointments at all hours. I did 1 in the morning for a nurse. So, and I'm good with that. I'm completely fine with 1 in the morning. So completely flexible time. So as Leo said, the big bottom lines of the study were, come up with this less sugar, more plant parts, which are not identified sometimes, is we need to not peel our vegetables and fruits when we don't need to. We need the structure of the plants. And less red meat, possibly less meat. Well, that's alcohol did not come out in the study, but I'm adding that just for social. Okay, and there's me. So we're done. Ooh, 6 minutes.
Great.
All right.
Thank you, Cheryl and
Thank
Leo.
you, Cheryl.
Thank you.
So that was wonderful. Great to be on time here. So we do have a few minutes for questions. I'm going to ask a question I've got here about probiotics. And maybe this one's a little more directed to you, Leo. But I mean, what probiotics are recommended that are currently available on the market?
You know, to give you the short answer is that there is not any particular one is recommended. There are studies have been done with probiotics, and Crohn's disease have been very disappointing. Unlike ulcerative colitis, there are some studies done with VSL3 and E. coli Nissl. Those are VSL3 is a cocktail of 8 different probiotic species put together in high amounts. And as we always said, and we had several conferences even at Yale, where we had every 3 years a probiotic conference, and I always did the IBD part. And so it was a very good company and very critical audience. Basically came to the conclusion that you can only recommend probiotics in the studies that they were done and in the amounts that they were published in. But a lot of the probiotics that are now over the counter are not even close. So typically I don't recommend probiotics in the way it is because it's not enough, it's not studied, and people pay out of their pockets for it.
Thanks, Leo.
Get it from food, right?
Sorry, Sharon? Okay, go ahead.
Get it from food.
Yes.
So as you know, our main cohort study in Imagine has a very lengthy food frequency questionnaire that, you know, much of it is, you know, recall over the past year. What information could a lot of our research coordinators provide to subjects to give them reassurance that, you know, obviously this is a validated tool, but that there is useful information that comes out of this dietary questionnaire?
Cheryl, you want to take that one?
Sure, I'll take a stab at it. It is, yes, it is controversial. So it's important to see trends, and the big trends are important, and that would be meat. Do they eat meat? Start with that. What types, and how long has this been going on? So those trends are important to see what kind of flexibility the patient has or the participant has, and as well, what Amar used it for is for, I guess, for the fecal health protecting, right? So maybe you can answer that a little more, Leo.
Yeah, so I think the food frequency questionnaire will give you more like the dietary patterns, right? So the long-term things, and they have been very validated. So it's all about validation. I mean, if you, and this way we can compare things over the world, but as you also know, and Cheryl probably can chime in on that one, that people have cultural differences. So your food frequency questionnaire needs to be also being validated on, and is sometimes culture specific. So not all the foods are being eaten in India will be the same as one eaten here in Canada, in Western Europe. So there are challenges with that. It's the best we can do by coming and using a tool such as this, what everybody has been validated and agreed upon. I think that's the only way we're moving forward and finding dietary patterns. It's a long term, and then you have the 24-hour recall where we order 3 days that we're just trying to get a more a sense on what people remember, because you know, remembering things and remembering food what you is going to be an issue on multiple dietary intervention. Once you sit with these people like Cheryl did, and for 6 months, and remembering what you ate yesterday, the day before yesterday, it's really to have that information is really important. And that's how you get prospective information, much more valuable than only relying on what people remember from a year ago.
Thanks. So there's a question here, how accessible or where can you access the fermented inulin?
Yeah, so that's a really good question. It's a fermented inulin. For instance, Benefiber is inulin. So that's available here in Canada. It's actually available in health food stores. But there are other ones, that's the prebiotin that I've been using from a company, Beneo Orefti in Belgium, that makes it from chicory roots, sugar beets actually. It's available in health food stores you look, but some of those things have to be imported from the US. So it's not common. It's you have to look for it. And my patient have been in the study and wanted to use it afterwards, had to direct them to a website. But you know, as this field is evolving, I wouldn't be surprised if some of those products are becoming available. The problem is that somebody calls something a prebiotic, and this is a great so-called literally selling point. And said, for instance, oh, here we got bread that contains prebiotic. Well, guess what? It's also in the bread is yeast that will basically eat the prebiotic before it gets to your stomach. So that's BS. So there's a lot of BS out there, unfortunately. So people just look, oh, prebiotic, it must be good. Well, it's the same thing with probiotics. It's only be good if it's well studied. And that's what we're trying to do.
Thanks, Leo. Another question, I guess it was in relation to your answer around probiotics, how you were able to consume a lot of that through your whole foods. Is that true of prebiotics as well? Do onions, garlic, artichoke, exactly, for example, or are supplements effective?
Yes, so if you would eat the amount that we use in the study, the 15 grams of the inulin, oligofructose enriched inulin, you would have to eat a lot of those foods in order to get the same effects. So those supplements are highly concentrated, but it's also true that our diet is devoid, Western diet devoid of those prebiotics. So yes, it is to be recommended that you eat them as well, but the supplements will bring you into the so-called area where you said, this is definitely enough. So does everybody need the supplements? That's something I don't know. We in our dietary studies, we're trying to incorporate that information and get a hand on how much people actually were eating. So I don't know if Cheryl has any information about the anti-inflammatory diet. Did you get information about prebiotics when you were doing this?
I did investigate levels of prebiotics, but it came down to basically eat as many foods as you possibly can containing prebiotics, because it wasn't good information necessarily that we found. So, like you said, it takes a ton and ton, and so it's not.
Well, an interesting phenomenon is if you eat prebiotics but you don't follow the anti-inflammatory diet, and we have done this in the HLA-B27 transgenic rats, that its effect of the prebiotics goes completely away if you don't eat a healthy diet. So that we don't have a diet full of omega-3 fatty acids, that effect can be abolished. So in other words, your background diet is likely more important than your supplementation with prebiotics.
And so I think this is going to be our last question. We are running just over time here, but maybe if you could speak a little bit around the role that increasing resistant starch in the diet might have on improving IBD symptoms.
That's a really good question. So resistant starch is one of those prebiotics, and I think a resistant starch is among the prebiotics the one with the highest amount of butyrate production. So if butyrate is your goal, and butyrate is, we think, well, that's correlating with effects in like I showed you in ulcerative colitis, then that resistant starch is something that should be looked at. All the other thing about resistant starch, it doesn't seem to give as much bloating as some of the other prebiotics have done. So we also begin to realize that not every prebiotics, and just like probiotics, not every prebiotics is the same regarding its effect on health and regarding its side effect profile. So resistant starch is an interesting one. We currently are embedded, we got permission through the Western Microbiome Foundation to study postoperative Crohn's disease prevention after surgery. And Karen Matzett and I have composed a symbiotic where we're taking probiotics and prebiotics cocktail, and it contains resistant starch, but also contains galacto-oligosaccharides. So it's a cocktail of them, because we really want to go for an effects side, like that. This is, so in other words, there's no golden boy here. It's not something like, okay, that's the winner. Those studies have not been done to compare one prebiotic versus another one. But resistant starch is a very interesting one.
Thank you. So again, thank you to Dr. Dieleman and Cheryl. We really appreciated your insight into the very complex world of diet and microbiome. And thank you again to all our participants. As I mentioned before, please complete the survey that you will be sent in the next little while. We really want your feedback. And again, thank you to all our sponsors, and of course CIHR, who's our major funder of the Imagine Network. So all the best to everybody, and hope to see you next month for our monthly webinar series. Take care.
Thank you. And thank you, Cheryl. And thank you, Ida.
Thank you.
Thank you.